We've been doing adverse reactions, and this is what our fourth season.
Amazing.
So, David, do we have a theme?
Everything is so much more complicated and interesting and more interconnected than we ever think it's going to be.
We've really kind of strayed from traditional toxicology in this season.
It's true. We have been expanding the reach of what most people think of as toxicology because one of the things I love about this discipline is that it is a necessarily applied science. And that means it touches basically everything, all the other sciences.
Have a listen.
Next, adverse reactions. Pipping the Scales with Zebrafish.
We wanted to look at benzoyl-pyrene, which is a pH and has been classified as carcinogen. And we wanted to see what the toxicity of it in zebrafish. So we did an exposure, we got the dose response curve, we found a NOEL. We found that they caused swimming behavior changes at 5 days. So then we're like, okay, what if we raise those same fish up into adults? Will there be anything that physiologically wrong, or is there anything that's behaviorally wrong? So we looked at their BMI when they got to adults, and they were a little bit on the high end. So zebrafish, we can actually do fitness tests too.
We're here today with Lisa Truong, PhD, MBA, SOT full member. She is an associate professor at Oregon State University and deputy director of the Sinnhuber Aquatic Research Laboratory. Thank you so much for being on the show with us today.
Yes, welcome.
Thank you for having me today.
Now, you have letters behind your name that we don't often see. MBA.
Yes, I got that a little bit over 2 years ago.
Why?
I decided to go a different route than most individuals. I graduated my PhD and went into the computational world in regulatory environment of the US EPA and decided that I didn't want to be that far removed from the data and decided I wanted to go back. However, I didn't want to have all the stresses and pressure of having a large lab and decided that, okay, I want to run a large lab. And so I work with Dr. Robin Tangway here at Oregon State, and she's very successful and a pioneer in the zebrafish world. And so together we run a large facility. And so I've learned early on that I'm more equipped for leadership experience. And every time you go into academia, you're expected to run a lab and have no training. You're running a small business, but you have no training at all. So I decided to go back and get that training to run the small ship, which is about 40 people here.
That is smart. That is super smart, because if you're going to take the classes anyway to learn how to do your job, you might as well get the credential with it. And that's not just something that you learn in graduate school.
No, it's not a skill that we're taught. It's not a skill that we're expected to understand. And we just picked up somewhere because from role modeling, because no one's trained for it.
It's an interesting example of the further you go in academia, the less each phase prepares you for the next thing you will do. Like you say, these are small businesses. Running a lab, and even an academic lab, is a small business. But there's a disinclination. People who are in the sciences, generally speaking, from my experience, are not as interested in all of the nuts and bolts and money side of things because they're like, oh, I just want to do the science. I don't want to have to worry about budgets and all of these other things. But then all of those things are really important.
The reality hits, right? When you get in and you realize, I need to budget, I need to understand the operations of everything. I found that having project management classes just wasn't enough. That's when I was like, okay, I need to think bigger, and got my MBA recently.
Well, let me take a step back for just a second. So you meet my mom at a party.
Lovely woman, Anne's mom.
Yes. So what do you tell my mom about what you do?
What I normally describe to people is that I look at how chemicals interact with the environment or human health. And I use a model, which everyone doesn't know much about, a fish about 2 inches wide that surprisingly shares a lot of similarities to humans. And the first reaction everyone always has is, it's a fish. I don't swim like that, or I don't breathe underwater, and we don't have gills. And I always explain that zebrafish and humans have about all the same major organ systems, like the brain, the pancreas, liver, and even though we don't have gills, we have a lot of the genes that developed into the gill. And for us, our ears, fish don't have ears, but they have osit and vessel, which is similar to our ears. And all of the genes are conserved between fish and human. And what we do is we take advantage of that in this fish model, because a single female can produce 200 eggs. And those eggs develop from a single cell to a larval fish is swimming around in 5 days.
So that's really cool that you could basically just see the development under a microscope. For the endpoints that you're looking at, a lot of the things that you're looking at is how chemicals affect the developing embryos, right? You're growing these eggs in water outside of a body. We grow inside of other humans. Does it matter that there's that difference in the way that the embryos develop?
So there is different ways of exposure. What we do is the fish eggs have a shell, just like an eggshell, but it's a little bit more palatable and there's called chorion, and we remove that. So we try to encourage as much interaction with the chemical as possible. And honestly, development is the most sensitive life stage. So we basically say you're swimming in this without the chorions, and you're swimming in this for the next 5 days during development when you're going through every single signaling pathway possible. And we challenge you to come back out physically, morphologically normal, or seeing if there's any neural toxicity or behavior with it. And do they swim differently? Do they respond to sounds differently? And are there long-lasting effects? If you take them out of that chemical, you raise them in clean water, will there be long-lasting effects that impact their social, their cognitive ability, learning and memory? And so there's all these different ways to look at their impact during development.
You mentioned a lot of different tests that you can do for neurological development. And I'm really curious, what do those look like for fish? How do you do a neurology test on a fish?
There's lots of different ways. What we do at the beginning, we wanted to see if there's any neurobehavioral differences first, because we don't know if it's behavioral. So, for example, we do a test where we turn the lights on and off. For fish at that life stage, when the lights are on, they freeze because their natural behavior is they think predators might eat them. So they're going to tense up and just stay there. In the dark, they swim around looking, foraging, and kind of doing their own thing. So we can test if the chemical exposure during development, if it caused them to not swim as much in the dark, or they swim too much in the light, that they don't have this fear response or preservation. And that's just a behavioral change. We don't know if it's neural behavior until we check if their muscles are impacted. Like if they're swimming too much, is it because their muscles receptors have been impacted, or is it that their eyes aren't functioning? They don't see the light. They just think it's the dark all the time, or they just think it's light all the time. That's why they're not swimming. Once we can eliminate those two physiological factors, then we can say, okay, it happens in the nervous system. Something happens during that point, and we can pinpoint by something we do, which is adjusting the time of exposure that they're in a chemical. Expose them only after the nervous system is developed, or only during the period of time when the genes of the nervous system are present.
In my line of work, we'll have to do a zebrafish assay or a fish tox assay, this, that, and the other. So how do you focus it down into a research question or a path?
We do a lot of different things. I did my PhD developing the high throughput screening platform, developing the assay and automation to deliver these embryos into 96-well plates and adding the chemicals and doing this just a very general assessment. But the reason was to help prioritize chemicals. With all the chemicals out there without safety information, we wanted to help prioritize, especially since their focus that they had was only on trying to do in vitro assays and the high throughput. It was lacking the behavioral component because it's a dish of cells. It's only one type of cells. There's no interaction. The original focus was to do for every single chemical and get all this comprehensive data on every chemical to make a decision of which bins to go into. So there was one assay we started with, which was one behavioral test. And that was the one where I was explaining the way you turn the lights on and off on them. That became the trigger to say, okay, if there's nothing going on, or there is something going on, they get into different categories. Then we started going in and looking at certain classes. We did flame retardants, we did PFASs. At Oregon State, we have a super fun research center, and we work on polycyclic aromatic hydrocarbons. And so we've tested the largest library of those too. So we are focused on classes, and we've done endocrine disruptions too, where we find the ones that are causing effects during development. And then we go down to Noel and see if there's any long-lasting effect and behavior. And so we kind of have a tiered approach.
It's amazing that you can do all this really fast, because you mentioned 5 days, right? Development time. That's extraordinary. And maybe this is a silly question, but why zebrafish and why not other types of fish? Or are there other organisms that could be used for this sort of thing?
There's a lot of different other fish species that others are using for different purposes. And there's other pathways that they're very conserved to. It's just the development time. And others is that they're not saltwater fish. Some of these other fish are saltwater. And so that makes the chemistry a little more difficult. Proteins and salts just change ionic strength and cause a difference in bonding and acts very different. And so we were trying to find a model that was not going to end up causing problems for the analytical side. Adding extra components like bovine serum has all the different proteins, and so then it's all the binding that was happening to the nanoparticle, and that added confounding factor because they would just precipitate out.
I know that nanoparticles have historically been kind of tricky to deal with in a lot of assays. How well does this assay or system deal with nanoparticles, and what are the complexities? Why is it the case that nanoparticles are more difficult to deal with in a lot of these types of things?
The biggest challenge for us when we got into the world of nanoparticles, which is what I worked on primarily for my PhD, was that the material that they were synthesizing was such small quantities because it's a nanoscale. So to make that one batch was so precious to them. And then because of the properties that they were using, sometimes it would repel water. It wouldn't go in solution, or it would bind, which is what it's designed for. It binds to salts and it would be binding to proteins. And so the results were very much uninterpretable, because in the cell cultures, they'll be like, oh, there's no toxicity. Then that's because it's bound and they precipitated. When in zebrafish, we saw a little bit of that because we had some salts, but not as high concentration. The salinity wasn't as high, and there's no proteins in our media. But that still caused a little bit of a worry. So what we discovered was zebrafish during development, they can actually develop in ion-free water. And so we eliminated that completely. We can actually see their full potential of toxicity in this model that wasn't foreseen. And we're doing green by design, working with a lot of chemists working really closely, and they would put new different ligands on there because some of their ligands were not toxic, but when you combine them together, they would be toxic in this situation.
Are zebrafish inbred? Do you have to worry about mating?
We can. It's ironic. Zebrafish was discovered as a model just for development in University of Oregon. And at that time, they created a zebrafish line called AB. And the story is that they went to pet store A and then pet store B, and they bred them together. And that line bred to be pretty much an inbred line deliberately.
Wow.
Since we had that when we first got into the field, we wanted to get the most wild type fish genetic diversity that you can find. I think it's the tropical 5Ds. We got it from the fish store Tropical. And we went to India and they brought fish back and they bred it to be outbred. And so we do have to outcross them. We actually have some studies where we're doing gene-environment interaction studies where we're looking at the genetic variability. Why would a pool of animals, they exposed to an EC50 concentration of a chemical, why is 50% have an effect and 50% doesn't have an effect? Is it the gene? So we did some sequencing and we found that between siblings from the same parents, there's about 5 to 7% differences. And so we have a line that's really outbred and the other one that's really inbred. So in our facility, there's about 37 lines that we have available to us to do these different types of testing.
Incredible. Well, and then there's this other layer of complexity because not only is water complicated, but you've got all these different strains, but then also you have microbiomes that can affect your toxicity studies. So can you tell us about that?
We work with a group here on campus, Dr. Tom Sharpton, and we develop a way to actually assess the microbiome of zebrafish looking at their poop.
As one does.
When they're younger, they don't have enough for us to collect fecal samples. And so they can actually, they're really good, talented at this. They can microdissect out the gut of the 5-millimeter fish.
Amazing.
So I used to think I was pretty cool because I could pick a rat nose. I've never thought about picking a fish gut for poop.
Yeah, very talented at doing that. We found that the gut microbiome after exposure during development, this was actually benzylpyrene or PFAS too, we found that their microbiome changes and it's correlated with their behavior profiles, the responses that they get.
Does this mean that we should all be drinking more kombucha, eating kimchi, that sort of thing?
I don't think we know enough about that yet to actually be saying what is the best microbiome remedies.
It is interesting, though, thinking of the gut as having a really important role in a lot of disease that you might not otherwise associate it with.
We did some research with understanding how micronutrients is also important to our behavior and also the health of the next generation. If you supplemented or you remove certain vitamins out of the zebrafish diet, you find that certain, like either zinc or vitamin E, the lack of or excess of it can cause deleterious effects in the offspring. Lots of different gene changes, and looking at the microbiome, there also has those impacts.
So you mentioned behavioral changes and social behavior as well. Can you tell if a fish is depressed?
Oh, that's good.
I think you can. You can almost tell in the sense of zebrafish are social animals. Like in a regular non-laboratory setting, they are always in schools of fish. In our software, you can measure how far apart the fish are from each other. And which is like what they call the nearest neighbor and the shoaling cohesion. But they also, when we singulate them, We pull one fish out and isolate it. You can see what they normally do. They go into a new tank, they kind of patrol on the bottom of the tank and just hang out. And eventually they'll start exploring. You can look at that latency of when they start exploring or if they ever do.
Or they're just like, well, I guess this is my life now, alone at the bottom of this tank.
That was the best thing I heard all day.
Finding emo.
Finding emo. So how do you extrapolate some of the neurobehavioral things and turn that into something that the public cares about?
One of the studies that I could talk about is that we wanted to look at benzylpyrene, which is a pH and has been classified as carcinogen. And we wanted to see what the toxicity of it in zebrafish. So we did an exposure, we got the dose response curve, we found a NOEL. We found that they had caused swimming behavior changes at 5 days. So then we're like, okay, what if we raise those same fish up into adults? Will there be anything that physiologically wrong, or is there anything that's behaviorally wrong? So we looked at their BMI when they got to adults, and they were a little bit on the high end. So zebrafish, we can actually do fitness tests too. So we had these little water treadmill.
Amazing.
So we can see how much oxygen they're consuming.
Incredible.
These fish that were exposed only during development of 5 days, they consume a lot more oxygen. And so we're like, okay, so what else is impacted? So then we looked at their learning and memory, and they weren't as sharp. Like it took them longer to learn. So we developed these little shuttle boxes where zebrafish, to condition them, you can feed them food. There's negative conditioning to fish that we can do in a high throughput manner. And it's like a mild little tap to them. And they kind of go to where they need to go. So we put these fish in these tanks that are divided where when you turn on the light, you want them to go into the light. And they have 8 seconds to go to the light as we condition them multiple times. And those fish that were exposed only during development, they never caught on. And then what we did was we took those same fish and we developed these assays where basically it's a drive-in movie. You're in your own box by yourself, and we show them a video. We first show them a video of their friends. And so we have a camera right above to see how close they hang out next to the screen, because they're like, my friend or not. And so they swim right next to it the whole entire time. Then the video switches to a predator that they've never seen in life because they're raised in captivity. It's a predator and they dart. We see how fast it takes for them to leave. Because they saw their friend, we know their eyes are working. So there's no pheromones related because it's just a video. And zebrafish can only see size. Even if it wasn't a picture of a predator, it's just a size of their dot growing. It seems like something's coming at them. So you can see how much time they spend away from the screen. Then the last test in that same assay is if I came into the room and I popped the balloon, everyone would kind of startle for the first time. If I keep doing it, you guys will be like, knock it off. That's a no. The fish do the same thing. They have the same response of the lateral line feeling of that. And we can test that for zebrafish. And what we expect is that the first tap, they be a high response, and over time, they habituate and ignore you. And what we found from the study is they performed poorly in all those behavioral assays when they were only exposed during development. So it's kind of, we wrapped it up to like they had learning and memory problems and maybe high anxiety, because they never habituated to that response. And so then what we wanted to see is if this is transgenerational. We wanted to see if their children had this effect. And they were never exposed at all, except for their parents had been exposed. And the trend was consistent. The BMI was back to normal, but they still had fitness deficits and they had a lot of learning memory issues. And then we did grandchildren, and the effect's still happening. We went 5 generations. And a lot of the adult behaviors are not as strong, but there's still some effects where we can see that they have anxiety and their learning memory is not as acute as before.
2 immediate thoughts. First is very relatable, being anxious and overweight. Totally get that. Secondly, the description of all these tests are adorable. I very much want to see this because it sounds awesome.
I want to be a fish friend.
Yeah.
So you're at EPA, you're this brilliant graduate student from your lab, brings this to you, you're a regulator. What do you do with that?
That is a challenge right now, trying to understand what to do with the data as an adult for a fish.
How do you take what you've got there and translate that into saying, we're not regulating these chemicals enough? How do you take this and translate this into something that isn't going to just freak people out, be able to adequately convey the risks of eating too many really well-done hamburgers? Because that's really what we're talking about with PAHs, right? Is well-done hamburgers.
Yeah, so Dr. Tang Wei is who I work with really closely. She's working with the different groups on trying to figure out how to apply new Alzheimer's methods to understand how that brings value. And so what we're proposing is really that zebrafish is going to be the first screen, the tier, and that if these assays come up and these are hit, then follow them up. And what the studies will give us data and be able to corroborate and get more information to say, this chemical is what we see in zebrafish, and we see the same trend in these neural cell lines. Or we have to go to rodent studies, but trying the 3Rs and trying to move away from it, trying to find alternative assays to actually supplement the information based off what we find. And so not necessarily saying that zebrafish is end all be all and that we found the answer. It's this is the start. We have some inclination of what could be the concerns. And so what other assays are available in this toolbox can we use to actually move us forward? He's hit it on the spot, and not everyone knows what to do with the zebrafish data yet, but they are concerned and they agree with the data. It's just how do you get into regulators to understand that? But right now it's cost prohibitive because we're just doing one concentration at the NOEL. But is it not enough to say that the NOEL has a long-lasting effect?
So the NOEL being a no observed adverse effect level.
Yeah, when they are being tested during development. But it becomes long-lasting effects that no one's been looking at. That they would have checked off, and if it was in any other assay, they would have checked it off and said, good to go. But we're not seeing these other effects. And so I think we're going into a world where we haven't even looked into those potential. And I think NIHS and other government granting agencies are starting to look into those potential impacts, but it's not clear how to deal with that yet. Like, how do you calculate a NOEL for adults?
Yeah, exactly. So since we are talking about adverse reactions, what was your biggest adverse reaction?
Oh, that's on the spot. I didn't actually think of one beforehand. I would probably say my biggest adverse reaction is actually being the first to get into grad school in my family, and that I didn't go into the stereotypical medical field in my family.
Wow.
I wanted to be a researcher and study toxicology. I came to Oregon State as a pre-pharmacy student. I got admitted to the program, and that's because the stereotypical tiger mom of mine was like, you need to do something that's not just gallivanting and save the world. And so I was saying, I want to study what causes the toxicity and not only sit there and count pills all day.
That's interesting. That is really interesting. I think that's the first one we've gotten of that nature.
I think that's really interesting from a parent right now who's trying to get their children to choose majors in college that will get them off of my dime. There's a balance of that. I remember telling my parents, yeah, I'm going to get a degree in biology. Yeah, I'll get a job. Oh, okay, sure you will. And then when I did, they're like, oh, thank God. Because I told them all throughout I was, and then I did, and they're like, oh, good.
That's the reaction I got. And when I decided not to pursue my pharmacy degree and declined it, that was the reaction. And then going into grad school right away. Because I did my undergrad faster so I can get into pharmacy school sooner because I finished in 3 years. And so I can start sooner. Then I decided a quarter before that I'm going to apply for grad school.
Amazing.
Good for you.
So speaking of the road not taken, another favorite question that we like to ask our guests is, what would you be doing if you were not doing what you are doing right now?
I would probably guess it's something to do with running some type of organization, probably, like in the medical field somewhere. I grew up really poor and I'm bilingual, so I speak Cantonese first. And so trying to help with them and going to doctors and trying to help them navigate everything. My parents were immigrants over from the Vietnam War. I was born here, but I speak English well and whatnot, but others didn't get that privilege, right? So parents didn't have all the access to things and don't understand how the society works and are taken advantage of all the time. Not that anyone's intentional, it's just they weren't informed. I think in an alternate universe, if money was not what you had to survive off of, that probably would be doing that.
That is awesome.
That's really cool. Thank you so much for coming on our show today. This has been a lot of fun. Really, really interesting stuff. Thank you.
Thank you for having me.
Thank you so much.
Next time on Adverse Reactions. Toxicology is a team sport. The science of working together.
With Dr. Steve Fiore of the University of Central Florida.
Most people don't realize teamwork is a new idea in organizations. Teamwork in the US really didn't take off until like late 70s, 80s or so. And even then, it wasn't really the way that everyone was doing something. Even organizations were very individually oriented. I think this difference between a more collectivistic orientation versus an individualistic one, it's not just in science, it was really part of the US.
thank you all for joining us for this episode of Adverse Reactions presented by the Society of Toxicology.
And thank you to Dave Levy at Maestro Studios,
That's Maestro with a 3, not an E.
who created and produced all the music for Adverse Reactions, including the theme song Decompose.
The viewpoints and information presented in Adverse Reactions represent those of the participating individuals. Although the Society of Toxicology holds the copyright to this production, it has
Definitely.
not vetted or reviewed the information presented herein.
Nor does presenting and distributing this podcast represent any proposal or endorsement of any position by the Society.
You can find out more information about the show at adversereactionspodcast.com.
And more information about the Society of Toxicology on Facebook, Instagram, LinkedIn, and Twitter.
I'm Anne Chappelle.
And I'm David Faulkner.
This podcast was approved by Anne's mom.